Hello, and welcome to Genominon's on-demand presentation highlighting global clinical perspectives, optimized variant interpretation with the Mastermind Genomic Search Engine. My name is Mark Keel. I'm the Chief Science Officer and founder of Genominon. And with us today, we have two excellent speakers who are going to highlight their use of Mastermind in clinical practice. Nisa Esen from Acibadem and Ariana Gonzalez from Héritas. By way of introduction, Mastermind is a genomic search engine, which is a comprehensive reflection of the evidence in the empirical medical literature, annotated for clinical and functional significance, used to drive clinical decisions in variant interpretation labs. Mastermind is the most comprehensive source for this information, reflecting coverage of more than 30 million titles and abstracts, more than 8 million full-text articles, including figures and tables, and nearly 3 million supplemental data sets. From this data, Mastermind, using its genomic language processing, captures the mention of all diseases, all genes, and every variant described by any one of these authors, no matter how they describe them. You can read more about Mastermind and how it compares to previous techniques by following the link below to our research report. I mentioned that Mastermind is a database. It is also a knowledge base comprising a number of previously published genomic associations. You can see a number of the parameters that we associate together through this indexing on the left. Genes and their variants, including copy number variants, all of the diseases and phenotypes that are mentioned in any one of these references, as well as the therapies that are used to treat those diseases and symptoms. And additionally, a number of categorical key terms that help variant scientists understand which articles are most relevant to their specific clinical questions. From these associations, powered by the evidence organized and annotated and presented in Mastermind, the variant scientist is able to see all of those associations prioritized according to the strength of the evidence in the Mastermind software. Mastermind has an intuitive search interface that allows users to take advantage of that genomic language processing used to index these references for their own search. Similarly, our philosophy is to show our work, and you see that reflected here, particularly in the lower section where we're actually displaying snippets of context where relevant information pertinent to your search is displayed in Mastermind to guide you in your decision-making process. Mastermind has been integrated into a number of third-party software providers shown here. We're continuously deepening and expanding our reach into these software providers and their offerings, as well as across the entire genomics ecosystem. If you have any questions about the partnership and the integration from any one of these providers or any other providers not shown, please don't hesitate to reach out. Lastly, I'd like to say that the information discussed in these talks today reflect the Mastermind Professional Edition, which is intended for clinical use, which is being highlighted in this webinar. And if you're not already a Mastermind professional user, I invite you to sign up. You can follow the link there and be connected with the software and see how Mastermind can help facilitate your variant interpretation in your clinical workflow. I'd like to introduce Nisa Esen from Acibadem in Turkey, who's going to go through a number of her own clinical cases, highlighting the use and utility of Mastermind in her practice.
Hi, I'm Nisa Esen. I'm the head of the bioinformatics department at Acibadem Labgen. Acibadem is the biggest hospital chain in Turkey and world's second largest healthcare chain under IHH Healthcare. Labgen is the genetic diagnosis center of Acibadem, and it performs a wide range of genetic tests, including cytogenetics, microarray, gene panels, exome and genome sequencing for common and rare disorders. In this presentation, first I will talk about why we use Mastermind, and then I will walk you through a case which we use Mastermind to rapidly uncover a new gene phenotype association that has not been listed on common disease databases yet. So we have been using Mastermind Professional Edition since January 2021 and have implemented it in our SNV and CNV search. And why we use Mastermind? It is comprehensive, and compared to other sources, it has an impressive number of variants, publications, and it also includes supplemental data, which is very important. And it is updated weekly rather than quarterly. With Mastermind's filtering options, you can quickly get the information you're looking for. Instead of drowning in countless numbers of publications, you can filter the results by ACMG criteria, such as inheritance or functional studies, or you can filter by clinical significance for diagnosis, prognosis, therapy, or even clinical trials. And you can also filter by genetic mechanisms, and under this category, for gain of function or dominant negative. During the variant curation, one of the main concerns of mine is if I'm missing something. And this worry keeps me searching for more, and eventually I spend a lot of time. When I get no result in Mastermind, I know that the variant I'm searching for has probably not been published yet. So at this point, I can stop my search and carry on. You can also create an alert and get notification if something comes up about your current search. Another reason for us to use Mastermind is they have an excellent team that supports and assists you through every step of your experience, and they respond to your problems very quickly all the time. Moving on to our case, 12-month-old male patient with craniosynostosis and some dysmorphic facial features was referred to us. He is the first child of non-consanguineous and unaffected parents. And we performed exome sequencing for the proband and detected no SNVs or CNVs in known craniosynostosis genes listed in common databases such as OMIM, ORFA, or Mondo. Before calling this an unsolved case, we decided to check rare truncating variants and high-impact missense variants regardless of the associated phenotype. And we detected a frameshift variant in BCL11B gene, which is a subunit of BAF chromatin remodeling complex. It is originally associated with two autosomal dominant phenotypes in OMIM and Mondo databases, which are immunodeficiency 49 and intellectual developmental disorder with speech delay, dysmorphic faces, and T-cell abnormalities. Both of these disorders are very early onset disorders, and they appear to be fully penetrant conditions. Our variant is a 22 base pair deletion in the last exon of this gene and removes around 10% of the protein. But it is located in the highly conserved zinc finger domain of BCL11B. Because of this, we thought even though it may not cause nonsense-mediated decay, it still can affect the protein. Allele frequency of the variant is also not present in NOMAD. So here we have a rare and potentially harmful variant, and we wanted to further look into the details of the gene. Here are the constraint matrix for BCL11B from NOMAD database. From the missense Z-score and PLI score, we can conclude that this gene is not very tolerant to variation. And here is the exome expression profile of BCL11B from GTEx portal. Our frameshift variant is in exon 4, so exon 4 is highly expressed in some of the regions of brain and skin, and this indicates the importance of this region. All this information together suggested that this variant has a potential to be relevant. So we performed a literature search with Mastermind to see if there's any human case or animal study that links craniosynostosis phenotype to BCL11B gene. We got 16 results. And the history chart here shows that the first publication is in 2015. Then to quickly check how many papers about diagnosis of a patient there is, we use the filtering categories here. And we filtered the results by clinical significance and diagnosis. And you can see the number of papers right next to the filtering criteria, even before applying the filter. The first article displayed here is the most relevant hit. So it's a study by Goose et al., published in 2019, that reports a de novo variant in BCL11B gene in a patient with craniosynostosis, which is exactly what we were looking for. Mastermind also highlights the match in the article. So even before going into the details and reading the paper, you can see here if you are on the right track. To see if there's any functional studies, especially animal studies, we filtered the results by ACMG tab and functional option. Here in the in vivo section, we see that 15 articles are linked to in vivo functional studies. So at first we got 16 results, and this piece of information indicates that almost all articles that links to our phenotype to this gene are animal studies. How Mastermind helped us here, as you can see just in a couple minutes, it revealed the information that the association between BCL11B and craniosynostosis has been investigated for 6 years. And there is a single case with isolated craniosynostosis with a de novo BCL11B variant. A majority of the papers are animal studies that shows BCL11B plays a critical role in craniofacial development. As the outcome of the study, we performed parent study for our patient and confirmed that this is a de novo variant, and genetic counseling was given to the family. And we explained that this is a possible new gene phenotype association that can explain our patient's condition. Currently, we are conducting further studies to confirm this diagnosis. I would like to thank Mastermind team for having me here. And I would also like to thank people from Acibadem, Dr. Yasemin Alanay from Pediatric Genetics, Labgen Director, Dr. Ahmet Yeşilyurt, and Labgen Supervisor, Dr. Said Tumer, for helping me with the case. Thank you.
I'd like to introduce Ariana Gonzalez from Héritas in Argentina, who's going to walk through a couple of clinical cases that she encountered in her workflow, highlighting the utility of Mastermind for finding evidence for copy number variants, both large and small.
So hello, everyone. My name is Ariana Gonzalez. I'm speaking in on behalf of the Héritas team, and I'll be talking a little bit about optimizing CNV search through Mastermind. In first place, I want to talk about who we are. We are a company focused on human health and wellness. We utilize genomic and precision medicine. We're currently based in Argentina with LATAM Bridge. What do? We have 2 units, a health unit and a wellness unit. Within our health unit, we use DNA testing to diagnose or predict risk for specific conditions like genetic disorders, rare diseases, cancer, and prenatal screening. And within our wellness unit, we also use DNA testing to obtain information that can be customized in wellness plans. So Héritas offers an end-to-end model for genetic testing. Within the health unit, we have 100% integrated solutions from end to end. In our core, we have the clinical genomic diagnostic assays. In the first step, we have the pre-analytical phase with genetic counseling. And here we have Dream24, that is our patient support tool. We offer genetic counseling, and it's carried out to inform, educate, and support individuals and families. In the second step, we have raw data and interpretation phase with the genetic analysts. And in here, I want to highlight that we are an interdisciplinary team that is conformed by the platform of data production, data team or data science, and the medical genetic platform that works together in order to reach excellence in our variant analysis and interpretation. So for us, each individual is unique, and we understand it as a unique way, and that makes us a better understanding than for each case. And in a post-analytical phase, we have genetic counseling, that is the interpretation phase, that is made by a genetic specialist. Since every step is consolidated within Héritas, within our laboratory, we generate and save the electronic health records that's product of the teamwork I previously showed. So this is our Héritas portfolio. We have different products. We have Vision, that's our non-invasive prenatal testing product. Focus, that is our clinical exome solution. Clear, that is our hereditary cancer solution. Chromo, that is our microarray solution. And OncoSense, that is our liquid biopsy solution. We count with 4 very important international quality controls, such as CAP, the European Monitor Quality Network, the UK National External Quality Assessment Service, and Seracare. And this is a picture of Héritas today, and without this working team, nothing would be possible. So I want to start with use case 1. Use case 1 is from Chromo, our array solution, and it's about a large duplication. We had a 1-year-old male patient under study for hypotonia, psychomotor retardation, macrocephaly, polycystic kidney disease, and ventricular septal defect. The high-resolution cytogenetics report reported an inversion of the Y chromosome. We performed a chromosomal microarray analysis, and the result was a 25.3 megabases distal duplication in the chromosomal region in the long arm of chromosome 1, more specifically in the last part. So as it affected the terminal region of the chromosome 1, and this region was big and contained a large number of genes, we didn't hesitate in jumping directly through Mastermind. We use the cytogenetic band, but it's important also to highlight that you could use the genomic coordinates for your search. We could easily and quickly obtain an exact overlap with our region. And it's interesting to see that also we can see regions that are contained intersecting with this region. And you can also go and tap the genes to see and compare and contrast if the genes you found in your region are the same that are in this region that you're matching with. So we found 49 articles, and since duplications that involve this region are extremely rare, we wanted to reduce this number a little. So we used the phenotype filter, and we filtered by microcephaly, and we found 7 articles. We found this article that was really interesting that talked about the pure duplications of the terminal regions and the delineations for a real trisomy. And this was interesting because it provided us vital information of the lack of actual data we have to really define the syndrome. And then we found this paper that it's from 2016 and talked about the detailed analysis of 26 cases of duplications and triplication syndromes. And this paper was really a breakthrough for this patient, because in general, carriers for these duplications are described as product of unbalanced translocations. So this really was a paper that helped us better understand the variant. It's also interesting to go and explore annotations and also see smaller or bigger CMVs that are not the one you're seeing as the highlighted, because you can always get more clinical data from them. So after issuing the report, the doctor confirmed that the karyotype delivery carried out on the patient was not really an inversion, but an addition material that was attached. And karyotype was also performed on the patient's parents, and a normal result was obtained. So we can confirm that our duplication was de novo. And we were able to find information quickly and accurately in relation to our variant and relation with the phenotype, and together with our algorithms and based on the ACMG guidelines, we classify this variant as pathogenic with no conflict. And the second case, it's a case that came from Clear from our hereditary cancer solution. And here is an exome resolution level example of how to use Mastermind. It's a 28-year-old male patient with a diagnosis of cerebral hemangioblastoma. We didn't have any additional medical history or family history of cancer. So the doctor was suspicious of a possible von Hippel-Lindau syndrome. The VHL gene was analyzed, and we detected a possible deletion in heterozygosity in exon 2. And the deletion was actually confirmed by MLPA analysis. So although our case was on course and ongoing, we wanted to really understand how frequent or how rare this variant was. So we searched directly for the deletion in Mastermind, and quickly and with no problem found the exact overlap with the region. And again, 86 articles were a lot to review. So we used the phenotype filter and filtered by cerebral angioblastoma and found 13 very interesting articles. But the one we wanted to highlight was this one that talks about the characterization of the VHL promoter variants in patients suspected of Fongibelin-Dos. And it talked a little bit about the frequencies of the different variants within the VHL gene. And only 11% corresponded to large or complete deletions. On top of that, only 17% corresponded to exon 2. So this really highlights the fact that sharing data and performing case studies is key or crucial to obtain a better understanding of variants and of the syndrome. So what are the advantages of using Mastermind? It enhances variant analysis on our daily basis. Using filters to direct searches, it's fast and easy to use, it's intuitive, and it helps in time management. And I want to stop here a few seconds because at the end of the day, it's not about delivering the report, but it's about delivering the best report possible for each patient. So for this, Mastermind plays a key role in our daily basis in order to fulfill this statement. Thank you.
I'd like to thank the audience for their attention and remind them that they're able to sign up for a 3-day free trial of Mastermind Professional by following this link below, or otherwise to reach out to us with any questions that they may have about the use or utility of Mastermind. Thanks again.